Right. Okay, good morning, everybody. Good afternoon, I suppose. Just gonna call our meeting to order today. We have some guests online and in person, and so as per usual, after our presentation today, we'll do Q&A both in the room as well as online. So please feel free to ask your questions wherever you are, wherever you feel most suited. Today, it's my absolute pleasure to introduce Dr. Ben Rotstein, who is an associate professor, colleague of ours in the Department of Biochemistry and Microbiology and Immunology at the Faculty of Medicine. His research his lab is named the Molecular Imaging Probes in Radiochemistry Laboratory. And, you can tell by his title today that we're going to be talking a little bit about chemistry, or maybe a lot about chemistry. That's okay. Ben makes it very accessible to us. Ben has been trained in Canada at Dalhousie and King's College, as well as the University of Toronto. He then trained in Boston at Harvard and the Brigham. His research has, always focused on modification and the use of chemistry, and now more recently in the ability to develop new tracers for imaging, which of course here at the Heart Institute is very important. And we're very excited about the progress that Ben and his lab has made. He's a very well-funded investigator and has been awarded many awards from things like NSERC and the Alzheimer's Drug Discovery Foundation, Early Researcher Award from the Ministry of Ontario and Innovation in Science. He's a panel member on CIHR and other mental health research organizations. And, the funding goes on and on and on. Let's see, what organization has been not funded by? And that is not only a testament to the excellence of Dr. Rotstein's research program, but also actually the applicability of what he does across all sorts of different, here we are at the Heart Institute, of course, we think about cardiac diseases, but Ben has been able to successfully apply what he does to Alzheimer's disease, mental health, other brain disorders, cancer, et cetera, et cetera. So this is something that, we value. And I think the ability to have that interdisciplinary sort of research program is vital for both understanding diseases that we know and love, like cardiovascular diseases, but also many others, as Ben will, I think, show us today. So thank you so much, Ben, for agreeing to speak, and we look forward to your presentation.
Thank you. Thank you for the very kind introduction. So this is a talk that's heavily focused on the chemistry side of the lab. So roughly, the lab, its aim is to enable clinical imaging research. And to do that, we do two very different things. On the one side is what we're going to be talking about today, where we figure out methodologies to label new molecules, discover new molecules, and evaluate them in preclinical imaging. And then what I won't be talking about as much today is preclinical evaluations tracers, which it involves target identification, working with people like Katie and other people who have a better understanding of the biology than we do in order to identify what's the right target, how do we address it, how do we interpret what our imaging is doing. But today we're going to focus on methods. So almost all the work that we're going to talk about today is part of our NSERC projects, and focuses specifically on Carbon-11 for positron emission tomography methods. So the goals are molecular imaging and PET, and there are many different modalities we can use to do molecular imaging. You can do molecular imaging using a contrast agent for any type of imaging modality. But there are specific advantages to doing PET. And that is that it contains all the imaging properties you would need for quantitative clinical safe imaging, including it's dynamic, it's fully quantitative, you can image deep tissue, so you're not restricted to a certain depth. It's non-invasive, it's safe for patients, and we have very high sensitivity. So we're talking sub-nanomolar levels of a signal that we can detect using a radiotracer, which is in stark contrast to things like MR or CT, certainly an ultrasound. The way that these work is that we have isotopic tracers, which means that we substitute a stable nuclide within a molecule for an unstable one. And it's unstable in a specific way, and it's that when it decays, it gives off a particle called a positron, that then travels a short distance, undergoes an annihilation event, and gives off these gamma photons, and that's what we detect. So the properties of carbon-11 are actually quite well suited for imaging. Here's our workflow. We produce the radionuclides. We have a cyclotron on the main floor of this building. This is inside the shields. Then we do our radiochemistry. This is what we're going to be talking about mostly today. And then we'll show a little bit of our preclinical imaging today as well, but it can also be clinical. So the workflow is all in one building. That's why the Heart Institute is so special for us. Okay, so the focus for my lab is largely on carbon-11. And this is a challenging isotope to work with, mostly because of the relatively short half-life. You may be more familiar with fluorine-18. That has a 110-minute half-life, so also short, but more convenient to work with. However, there are some advantages of carbon-11 specifically. It has very good imaging properties, so it's a pure positron emitter. It has a very low positron energy, which means we get very high resolution images out of it. It can be produced conveniently on the cyclotron, high specific activity. It's used for PET. These are a list of atoms on the periodic table that have useful positron emitting nuclei. So they have half-lives and imaging properties that are reasonable. And we're focused mainly in this top right corner. That's because these are the atoms that most often appear in biomolecules and drugs. Whereas these metals can be useful for attachment. It's mainly through bioconjugation. If you want to work with a small molecule and really do tracer level studies, you really have to be in this region. Carbon is unique among those in that it's present in all organic molecules. It's present in nearly every biological molecule. That means you can do real isotopic substitution and be able to study molecules in their native state. The challenge with any type of radiochemistry is that you're working with elemental oxides or hydrides. We produce these as single atom units. So carbon is produced as carbon dioxide or methane. They have one carbon in them. And that means that if we want to make a complex molecule, we have to form a lot of bonds really quickly. Same would be true of nitrogen or oxygen and also fluorine, which means we need efficient chemistry that can take these C1 or single atomic synthons and turn them into complex molecules. So that's really the challenge that we've taken on. Carbon-11 is widely used in PET, even though despite the short half-life, it's probably the bulk of tracers that are used clinically are labeled with C11. Here's a list of several of them used for various applications. So this is raclopride. We produce this here for use at the Brain Imaging Center for imaging dopamine T2 receptors. We use PIB. We also produce this here now. This is for imaging beta amyloid plaques, primarily in Alzheimer's disease, but also we're going to start some studies in cardiac amyloidosis. And then methionine is used for cancer imaging, specifically glioma. And then there's several other molecules here. Most of these we don't produce, but you can imagine they may be useful for various applications. So HET is one you may have seen here at the Heart Institute, very important for cardiac innervation imaging. What you'll notice is these are all labeled as methyl groups, meaning they have this CH3 group sticking off of a heteroatom. They're all labeled in the same way. So we take carbon dioxide from the cyclotron, we convert it to methane, and then iodomethane or methyl triflate, and we react it with a heteroatom precursor, and that engages in methylation. So we make methylamines, methyl ethers, methyl thioethers, or methyl esters. And that's all fine and well, but there are some limitations to that. If you want to be doing isotopic studies, then the molecules have to have a methyl group on them, which many of them do not. It's difficult to purify these methylation reactions, although it's certainly practical. And the molecules just simply, they look kind of similar. So what we're trying to do is instead of working with iodomethane, we want to work with carbon dioxide directly from the cyclotron. So we'll save some time on the production of iodomethane, meaning our activity yields will be higher. And we believe that because it's in a higher oxidation state, we can do some more complex chemistry this way. CO2 fixation is not terribly novel. Some of the earliest tracers with C11, so there's actually, I was looking it up a couple of weeks ago, and some of the earliest studies are actually from the 1930s. This is far before anybody dreamed of a PET camera. But taking carbon dioxide, reacting it with a Grignard reagent, these, for reference, were discovered in the 1870s. You can make acetate, and this is actually a tracer we use at the Heart Institute to image oxidative metabolism. Okay, so we can do useful chemistry with this, but the molecules tend to be pretty simple because these are very harsh conditions. And there have been advances over the years to have milder conditions, and be able to actually build up complex molecules through multi-step syntheses, these can be really challenging to do. So this is a molecule called PHNO, which is used for imaging the dopamine T3 receptor in the brain. So what our goal is, let's work with carbon dioxide, let's build up complex molecules, and let's see if there are ways we can do that more efficiently. And in milder conditions, we can actually start to do some really more interesting stuff in molecular imaging, what's been done before. Roughly, this is our strategy. This is some work I was a part of before coming to Ottawa, where we can take amines, we can react them with CO2 directly, form these kind of ionic carbonates. We can dehydrate them, and this is the key intermediate that I want to highlight. This is called an isocyanate, and from this isocyanate, we can react it with various molecules. These are amines or alcohols. We can make heterocycles, carbonates, ureas. That all works very well. This is a pretty fast two-step synthesis. This can take 1 to 2 minutes in total, and you end up with linking pretty complex molecules together. So the idea is we're going to make these types of molecules starting from through an isocyanate, starting from carbon dioxide. And this can be useful for discovering carbon-11 radiotracers, and can also be useful for discovering fluorine-18 radiotracers, if that's what your ultimate interest is, if you want to go a commercial route. So here's an example for this URB597, which is a serine protease inhibitor. So this specifically actually inhibits fatty acid amide hydrolase. And that works because of the carbamate in the center there. So the idea is, well, let's radiolabel that carbamate, and then our label will be stuck onto the enzyme after hydrolysis, and that works out very well. So you do the CO2 fixation method, you come up with carbon-11 curb. This is used in clinical research at CAMH. And then we thought, well, people want a fluorine-18 tracer if they don't have the expertise in carbon-11 chemistry, or they want to use it off-site. And using CO2 fixation, we simply screened a number of alcohols, a number of amines, and came up with this follow-up molecule, 18F-DOP, that's actually more sensitive than curb and contains F18. So you don't need to label it with F18 to begin with. We started by labeling it with C11, but then we found out, okay, well, yeah, we have a fluorine there. We can evolve the method to make F18. So not only is C11 a useful compound for imaging, but it's also a useful compound isotope for molecule discovery. And those are the types of methods we think are most powerful with carbon-11. That's where we have a major advantage over F18, where synthesis of precursors can be very time consuming. And so those are the kind of methods that we want to develop with CO2. Specifically, having established carbamates, ureas, heterocycles, we want to focus on amides. And that was one of the first challenges that we took on coming into the lab. So a little history. So before coming to Ottawa, this was a spurious result that kind of got me thinking for a while. So we were in one of those types of tracer discovery methods I showed on the last slide. We were working with these benzyl piperidine molecules. And we were linking them with nucleophiles. These were electron deficient alcohols, and we were using this kind of CO2 fixation method. And they would work very well, or well enough, with the 3,4 isomers. We'd take those forward and see if we had an enzyme inhibitor that we could label that way. The 2 isomer never worked, and we didn't really know why. We had some ideas. I filed it away. We decided not to pursue it because that was not the goal of the project. The goal of the project was to come up with the tracer, and the 3 and 4 inhibitors worked well enough. So file it away. Something else is going on. Fast forward a few years later in Ottawa, and now a student wants to conduct a reaction like this, where instead of making a carbamate through an isocyanate, they're going to make a carbamate by reaction with ethyl iodide here. And their precursor molecule was complex, so I said, okay, why don't you use a model compound? And from the student's perspective, it was any random model compound, something we found on the shelf, 2-phenylpiperidine. From my perspective, I placed this on his bench and said, use this molecule. It'll be interesting. And it was. So the reaction he had intended to run worked fine and well. And then I asked him, add some acid to it. He did. And all the product converted into something else, converted into what I really wanted to see, which was the amide. So here we're doing a carbon-carbon bond formation, which is much more challenging than a carbon-nitrogen bond formation. It's taking place fast in acid, compatible with all our C11 chemistry. And it's actually the major product in this case. So this is an interesting method. We're able to make the amides now. They're intramolecular. That's fine. Let's start. At the same time, speaking with some colleagues from Vienna, they were interested in this molecule called DPQ. DPQ is a poly-ADP ribose polymerase inhibitor. These are used in cancer. There's some clinical drugs like olaparib. Essentially this inhibits DNA repair. And they wanted to study this molecule because it was in a clinical trial at their facility, and they believed that they might be able to use it for imaging or at least study the pharmacokinetics of this. So it just so happened that Verina's student Marius was finishing his PhD at the time, came over to Ottawa to train with us for C11, and we set this out as a goal. Where else are we going to label this? Well, there's no fluorine on it, there's no methyl groups, so the logical position for us was, yeah, we can label that amide. We just need to figure out the right method. Marius did some initial screening, suggested you need to use a Lewis acid. On the previous slide, I showed you Bronsted acid cyclization. So we started with the Lewis acid method. And here's what we found. So starting with an amine, CO2 fixation, we go to our isocyanate, and then he treats it with aluminum chloride as his preferred Lewis acid and gets nowhere. So this is all symmetrical products, useless, not interesting to us whatsoever. So we realized that the problem here is that the isocyanate is unstable. With the aluminum chloride, we're activating this. We have an excess of our amine left over, and that's simply going to react and form these kind of symmetrical products. So instead, what we need to do is what we did in our little earlier discovery, which is mix in a Bronsted acid, and that's going to protonate out any excess amine. So we do that. We form the Lewis, the isocyanate now using Bronsted acid dehydration. Then treat it with the aluminum chloride, and in a single addition, 40% RCY, we're able to make this lactone. So that's pretty exciting. This is a bit of a reverse from what we did before, where now we don't have the cyclic amine, so they're a little bit more challenging. Marius went ahead and did a scope. These are all fairly robust molecules, and as you might expect, the electron-rich substrates work better than the electron-deficient ones. But you can do 6-membered rings, you can do 5-membered rings, and in the end, he ended up getting DPQ. Took this back to Vienna. They've done a little bit of PET imaging in some animal models, other models. They're very into organoids there. So they're taking that forward for imaging. So a nice little story of how we started making amides. In our lab, we were looking at something maybe a little bit more ambitious than making cyclic amides. We were interested in coming up with a cross-coupling method that would allow us to make these kind of linear amides that are more common in biomolecules. So this is reminiscent of any sort of peptide bond you might find in a protein. So we honed in on working with organozincs, and these are appealing because while they are organometallics, they're fairly stable. Some pioneering work by Paul Nichel in France has shown that you can make rather complex organozinc iodides. And also there has been some literature showing that you can react these with isocyanates, at least in a non-catalytic way. So here's where we're starting from. We're especially encouraged by this report in tetrahedron, where starting with an isocyanate and an allyl zinc bromide, then they can get this kind of sigmatropic rearrangement to form these amides. Without that specific setup, without the allyl group, they end up with these carbamates. And we thought, okay, well, we can intercept these types of intermediates, use a metal catalyst, intercept the organozinc, and then we'll be able to do 1,2 addition directly into the isocyanate. Maybe we can make this catalytic. This was actually the first chemistry project we started in the lab. And it was headed up by this guy, Mustafa Fouad, who's actually a resident of the Heart Institute now. He came by my office a couple of months ago, and I was thrilled to see him again. So here's the situation. We can make our isocyanate. We know that if we have a lot of water present, then we're going to end up with these ureas, which we don't want. We found, based on some initial screening, if we don't exclude air, then we end up with these carbamates. This is also specific to having excess zinc around. But if we use the right kind of catalyst, we can form an amide. And so I'm summarizing a whole bunch of work that Mustafa led to try to discover this. But in the absence of a catalyst, we see only diphenyl carbamate, not useful to us. With a palladium catalyst, which was kind of the leading approach, we end up with no amide and a bunch of diphenylurea. But if we use this rhodium catalyst, this rhodium-1 catalyst, we end up with a high yield of diphenylurea. So I still remember the day Mustafa ran into my office. He was an undergraduate, first time in the lab. He'd been there maybe 2 months. And he ran to my office, it works, it works, it works. And then he really went a little nuts and did a large scope of these compounds before we had an instrument to work with C11, so he had time. And so he worked and he made a bunch of organozincs and made a large scope of these. This is actually a catalytic method. We're using 2.5 mol% of the rhodium, and it works quite well. Very fast for the aryl zinc iodides, much slower for the alkyl zinc iodides, which we're going to see translated to the radiochemistry as well. At this point, Braden Mayer also joined the project, and they kind of split it up between half of the substrates for each of them. So translating it to C11, a few months later, we get our first C11 instrument, and we need to make this into a two-step method. So instead of starting from an isocyanate, we have to start from an amine, do CO2 fixation, and do this dehydration approach. And so there's two leading methods to do that. We can do what I showed you earlier with this phosphoryl chloride to do a dehydration. That's incompatible with our organozincs. Naturally, it's because phosphoryl chloride is releasing a lot of acid. So we resorted to this other method, which uses Mitsunobu conditions. That's this phosphate carbon-11 molecule debinding. In this case, these are compatible with organozincs, compatible with rhodium, and it turns out we actually get quite high yields if we use a specific super base called BEM in order to do this. So we get very high trapping efficiency and very high radiochemical yields with C11. Braden went about automating the method. This is a schematic he drew up that I like quite a bit. So it's like trying to directly connect to our instrument. We have our precursor molecule, our amine, inside in one reactor. We bubble the CO2 in, and then we add in our Mitsunobu reagents. We mix for a minute. We add in our rhodium, mix for another minute, transfer the whole thing into another reactor that contains the organozinc halide. From there, we can quench it, load it onto an HPLC for analysis or for purification of these molecules. Here's the scope with C11, and it reflects similar trends to what I showed earlier, where the electron deficient ones do not perform so well, but the electron rich amines, they do perform quite well. Hindered organozincs also struggle, but unhindered organozincs, they perform quite well. This works with alkyl organozincs, it works with aryl organozincs. We can actually make quite an interesting scope of amides here. The challenge is these were not perfectly soluble, these organozincs, so Braden ran into some troubles with the automation, but nevertheless, he was able to overcome those by just controlling the amounts quite well. We get very pure products. This whole method, starting from carbon dioxide from the cyclotron, takes about 20 minutes to make a radiolabeled amide after purification. That's very, very fast. Turbo joined the lab shortly thereafter, and her goal was to make a method that would not require organozincs, but use something that can be isolated and is solid, very bench stable. So she focused on using these organoboronates, working with copper, and she found a method that worked very well with conventional chemistry, was not compatible with her dehydration conditions. I'll just highlight these two rows here, where these Mitsunobu reagents are too basic for our copper catalyst. So we realized that this whole trying to mix the two processes together in one pot was really a challenge. And so where we went next was, well, let's get around a way to make isocyanates in a pure form without having to add all these different reagents. Because the more we add to the pot, the more sensitive our reactions are. While this is an encouraging method and we think we can eventually get this to work, we decided on a different approach. And that was to use these kind of pre-activated reagents. So this is called an aminophosphorate. Basically what it is, it's an amine that we've pre-activated using a triphenylphosphine, which is what we use in the Mitsunobu dehydration, but we've kind of combined them into one molecule that takes CO2, and instead of forming a carbonate, this directly forms an isocyanate. These are very reactive, highly nucleophilic, highly basic, but they're actually, before we had started working with them, this one is commercially available. Very few others had ever been reported in the literature, but we set about to try to make a scope of these, and we found out they're actually not that hard to make, and they're quite stable. You can do conventional purifications with them. So we were encouraged by working with these. The challenge is, because they're so reactive, once you go to isocyanate, it's difficult to stop it. So this reaction is called an acidic reaction. You get a double acidic reaction. So again, you make these symmetrical products that, again, we don't want. They're not helpful to us. While we're working on this, another group, Davide Odizio, Harris, he had started out working with these azide molecules and in one pot converting them to an aminophosphorate using this specific phosphine, and then those can react with CO2. He can make cyclic products. We thought it'd be more convenient to start with the preformed aminophosphorate. So this is his first project in the lab. He's now graduated a couple of years ago. So he also got very excited by the prospect of working with some of these molecules, and he went about making a large scope. These are quite interesting molecules. So you can make these carbamates that we've seen before. You can also make these thioureas that we hadn't seen before. These molecules on the right here, these are masked isocyanates. So essentially what he was doing here is trapping an isocyanate as a labile carbamate or a labile urea that simply when he heats it up, releases isocyanate again, and then he was able to make amides this way through either Grignard reagents or Sonic-Shearer coupling or copper catalyst. On the bottom here, these are all products made from new immunophosphoranes. So he's there mastered the art of making these things. And he came up with, again, with a pretty interesting scope here. Carbamates, again, these kind of masked compounds you could use as intermediates. He made this drug, regorafenib, that's an anti-cancer drug, as well as melatonin and a few other interesting molecules that we were excited about. This is all conventional stable isotope work. Moving on to radiochemistry, we need to optimize the conditions so that we would not get these types of symmetrical products. And it turns out it was actually a bit of a counterintuitive optimization. But usually we want to add more of our precursors in order to get the reaction to work faster. What Uzair found was that by chopping down the amount of immunophosphorane, cutting down the amount of DPU as well to make things less reactive in the pot, and then increasing the concentration of his interceptor nucleophile, he was able to get very high selectivity for the asymmetrical products. Here's his C11 scope. Again, similar types of molecules, carbamates, ureas, as well as some intermediates that could be used for acetylation or different kinds of bioconjugation. The bottom row here, these are more exciting molecules. This is an antiarrhythmic drug, experimental antiarrhythmic drug. This is CURB, which I showed you earlier, the fatty acid hydrolase radiotracer. And then there's this molecule that our colleagues in France actually were trying to make, flobenclamide. This is an anti-diabetes drug, actually, SIR1 inhibitor. They had been labeling it at this methyl group, but it required them 3 steps and over an hour to synthesize. We were able to make it in about 20 minutes in a high radiochemical yield. We went fishing around for what are some more challenging molecules we can make that might be interesting using these aminophosphorates approach, because we're very excited about it. And I saw in a magazine the structure of the NLRP3 inflammasome. I had no idea what that was at the time. And I decided to look into it. Yeah, one drug to rule them all. So this was the lead NLRP3 inhibitors developed in Queensland in 2015. And yeah, so it turns out there are many, many startup companies that are based around MCC 950 and coming up with other derivatives of this molecule, all based around this kind of thiourea, sulfonylurea pharmacophore. So the way this works is that NLRP3 is expressed in response to activation of IL-1R, as well as toll-like receptors, and then it forms a ligamer, this inflammasome. So you get these monomers that then all congregate to form this oligomer, where it has relatively few NLRP3s exposed, relatively few monomer, relatively few binding sites. What MCC does is actually opens up the conformation of that inflammasome, de-oligomerizes, so it's less active. Okay, while we're doing that, while I'm convincing Uzair to try this molecule, our colleagues at Michigan beat us to the chase. So they used conventional CO2 fixation. So that means that they start with the amine, and they're using this nucleophile, this sulfonamide. They're forming an isocyanate, and then using the DBAD, the Lisinobu conditions. And they end up making MCC 950. They have a very mediocre yield. They actually need to add more carbon dioxide to the pot, so that they end up with a very low molar activity, meaning that they have a high ratio non-radioactive drug to radioactive drug, which is poor for patient safety and for imaging purposes. When we went about it, we took the exact same approach, except we pre-activated our amine as aminophosphorane. That's a one-step reaction to make this stable in the freezer for months. And using very similar conditions, slightly higher temperature, we end up with a much higher activity yield, much higher percent yield, and much higher molar activity. So this is now a useful amount that we could use to take forward into imaging. The PET images themselves don't look all that exciting. All we see is that there's a ton of liver clearance. And we had these ApoE mice for a separate project. We were doing some matrix metalloproteinase imaging. So we decided to test out this molecule in these animals at the same time. So from the images itself, we can't see much within the aortas. We take out the aortas and we image them by autoradiography. We see that, yeah, indeed, there's much higher uptake within the plaque, which is as expected. And it seems to also colocalized where there's higher CD68 inflammation, a marker of inflammation. We want to test how specific this is. A lot of tracers that may be taken up with plaques are not so specific. They're just being taken up by a lipophilic environment. So the way that we do that is we pre-treat the mice with an inhibitor that should occupy the binding site. So we dosed them with 5 mg per kg of MCC950. This was, I think, the highest dose previously reported in the literature, so that's what we went with. And here we see what we anticipate happening is that this will occupy all the binding sites, and our tracer will have nowhere to bind and clear out. And that's the opposite of what we found, to our surprise. So the animal that was dosed with MCC950, non-radioactive drug, actually sees more than 2-fold higher uptake into the plaque. So why is this happening? There's a few possibilities. This is a simplified model of what how our tracer can exist. It can be in plasma. The plasma pool is in equilibrium with a tissue pool, and the tissue pool is in equilibrium with specific binding. In this case, we're suggesting that's NLRP3. So possibilities are that we're increasing the concentration in plasma. So maybe we're inhibiting metabolism by occupying the enzymes that are required to metabolize it. It could be that we're inhibiting binding to plasma proteins and therefore increasing the observed rate of transit from plasma to tissue. Or it could be that we're increasing this pool somehow. So maybe there's more binding sites. We went about and tested all the metabolism studies. There is no difference in the parent tracer concentration in either the animal strains or after treatment with MCC950. They both are very much intact. So that's not the explanation. There's no difference in, or very minor difference in plasma protein binding. This cannot reflect the 2-fold increase in tissue. So we ruled that out as well. Then we went back and looked at some of the other literature on what MCC950 does to cells, at least. And indeed, it increases the fluorescence of associated with Brett staining of active of NLRP3 monomers. Okay, so in cells, at least, when they're treated with an MCC950 to inhibit NLRP3 inflammasome, it deoligomerizes them, and therefore we have more monomers present, and that's what they're seeing. So our data are consistent with this. We haven't been able to go about and actually prove this, that this is happening in vivo, but it is evidence for this. This is reversible, so it does wash out over time. So that means that our uptake should also be reversible. And therefore, what we have then is a readout not only of NLRP3, but of NLRP3 inhibition, NLRP3 monomerization. Changing gears here, I'll talk about a more recent project in our lab. This is not published work yet, but this is something that pivots off of our aminophos framework. Here we're looking at these PSMA radiopharmaceuticals, and here are 3 of the most advanced PSMA radiopharmaceuticals in the clinic today. These have all been completed clinical trials within the last 10 years, and all 3 of these now have regulatory approval for use. So these 2 are for imaging. We have a gallium-68 radiotracer called PSMA-11, and DCF-PYL, which is an F18 PSMA radiotracer. The synthesis of this was actually devised in Montreal. And then there's this therapeutic molecule called Pluvicto that is very similar, and it's labeled Lutetium-177. These are useful for prostate cancer imaging, diagnosis, and therapy. What you'll notice about these molecules is that the left side of these is all just used for attaching an isotope, whereas the right side is always conserved. So these are all glutamate, urea, lysine, not a peptide, but actually a pseudopeptide, so they're connected by their N termini as a urea. So every one of these contains the same thing. If you look back at the medicinal chemistry studies, that's not a required arrangement. So the glutamate does contribute to high affinity, but the other side of it, they're only using this simply to attach another a linker to it that will allow them to radiolabel. So we're curious about this because there are some limitations in terms of the sensitivity of these molecules as well as the efficacy of the therapeutic. And that has to do with limited affinity of these molecules for PSMA and also limited internalization. And this is specifically important for the next generation of drugs like Pluvicto that are going to be used as therapeutics. The efficacy of the therapy is directly related to how close this gets to DNA. So we need these to be internalized. We need to deliver the radiation to the DNA directly. Therefore, having improved internalization, whereas the current ones internalize at about 17%, 20%. It's possible that we could discover molecules that are much better than that. I'm not going to say that the next PSMA radiopharmaceutical is going to be radiolabeled with carbon-11. It won't be. But the next PSMA radiopharmaceutical might be discovered with carbon-11, and that's what we're setting out to do. So we took our same approach, which was we take amino acids in this case, and we convert them to aminophosphorates. This is a two-step reaction that these hadn't been reported before, but we've developed. This has been reproduced by undergraduates in our laboratory. These are quite stable, so we've taken ones out of the freezer that are over 2 years old, and they still work. In terms of their analytical properties, they're still intact, and they're still reactive. So we came up with a way to make these in order to purify them. They're stable, and importantly, they also do not racemose. So here are some data with the glutamate aminophosphorane. So we're showing here that we can separate out L-glutamate from D-glutamate, the two enantiomers on HPLC using a chiral column. When we inject a mixture of them, we see two separate peaks. And then when we take this molecule, this aminophosphorane glutamate, and then we deprotect it and hydrolyze it back to glutamate, we end up with a single peak that corresponds with the L. So we're confident that these do not epimerize as the aminophosphorane. The reaction otherwise is very similar. So we take our aminophosphorane, we take a second protected amino acid, and now we have this combinatorial approach, right? This is the power of C11 and CO2 fixation, that we're taking two different inputs. These are both accessible. These are going to be commercially available. These can make one step from commercial products. And then we can mix and match and make a whole array of different compounds starting from CO2. So that allows us to make a wide array of compounds. These are all radiolabeled. This is a preliminary data set where we're still going about and confirming that every product is in high purity and is what we think it is. These are all N of 1. We need to go back and repeat these. These are what Hunter and Abhishek and a few other students in the lab have been working on recently. And some of the molecules, we've shown that they work with just about every amino acid that we've tested so far. We haven't tested histidine, but otherwise I think we've covered pretty much everything. This arginine works not protected. What we have found is that some aminophosphoranes do not work if they're not protected. So this tryptophan aminophosphorane, this actually forms a cyclic product, or I believe it's a cyclic product. It forms a byproduct. So we're not able to form this from tryptophan aminophosphorane. It may be possible to form it by activating the other side of it first. The yields here tend to be quite good. We're able to purify these products. We conducted preliminary PET imaging on one product. So this is glutamate phenylalanine urea. And this is imaging in healthy mice. So we don't have the xenograft models. That would be something we could test later on. But typically what you would see with a PSMA radiotracer is high uptake in the kidneys. And that's what we're observing here, primarily elimination through the kidneys. So this is the practical method for synthesis of new PSMA inhibitors as well as screening them. And now what we need to work on is developing that evaluation pipeline so we can try to find out which one of these have highest affinity and have good internalization into cells. Okay, switching gears to a totally different approach here. We'll still CO2 fixation, but now we got interested in this idea called carbon isotope exchange, where again, we want to think about how do we make this as simple as possible to discover new molecules. And that approach would be, well, start with the molecule you want and simply mix it with CO2 and then make the molecule, the labeled molecule you want. So these are identical, except for one has the radiolabeled isotope. The advantage of this is that you have available precursors. There's minimal purification time because you cannot separate these two out. Operationally, it should be simple. And this then allows you to use as a starting point for multi-step radiosynthesis. So if this happens so easily, then carboxylic acids are actually quite useful in synthesis. We might be able to make more complex molecules by a build-up approach. So the initial conditions that we were interested in were reported by Ryland Lundgren at University of Alberta in 2020. So what he did was he took ibuprofen and he had shown that with carbon-13, he can heat this up in the presence of cesium carbonate, DMSO, heat it up, and he can get incorporation of C13 to the carboxylic acid. So our first step was, well, let's replicate that. And we tried and it did not work with C11. So the reaction is too slow. The addition of cesium carbonate is probably a problem because it's releasing CO2 also into solution. So maybe it's causing isotopic dilution. This was not a functional condition. So we thought, is there another approach to this? The way that we think the reaction is working thermally is you get decarboxylation, you form an anion at the adjacent carbon, and that reacts with CO2. Is there another way we can go about this? And we honed in on photocatalysis might be an approach to this. So if we can initiate single electron chemistry to break this carbon-carbon bond, then maybe we can convert that back into the anion and reverse the reaction. So the CC bond will break easily with radical chemistry, and then using polar chemistry, we can reestablish the CO2 fixation. So it's kind of balancing two equilibrium. Here's the work with C13. Starting with this photocatalyst forces at IPN, this was done in collaboration with the London lab. They're able to observe good incorporation into ibuprofen. This is the catalyst that they used at the beginning of the reaction. They actually found that they were forming this catalyst during the reaction. So the substrate was actually degrading the catalyst into this other molecule. This other molecule, when we tested it head-to-head, actually was more reactive than the initial catalyst. So we get greater C13 incorporation if we started with forces at BN-BN rather than forces at IPN. That just has to do with this having a greater reduction potential than the starting catalyst. So again, we tried with C11, and using forces at IPN, replicating their conditions did not work for us. But then we thought, okay, well, why don't we start out with this more reactive catalyst? And indeed, this is work done by a postdoc in our lab, Maxime Munch, found that using this forces at BN-BN, now with a greater reduction potential, we can get efficient decarboxylation followed by recarboxylation. Does not work with forces at IPN, does not work thermally whatsoever. Here's a scope of molecules. So again, we're focusing on amino acids. We're interested in those. There's a bunch of drug molecules here that were just good substrates for this. Here's the preparative scale work. Maxime took this further. So the criticism here, using any sort of carbon isotope exchange, yes, this is very convenient. Yes, the purification is straightforward, but the molar activity is low. In some cases, you may want a higher molar activity product. Maxime came up with an approach for this. So rather than do carbon-carbon bond scission using the photocatalyst, now he's activating a CH bond, so carbon-hydrogen bond. And then from a very simple substrate, this is simply a hydrocarbon at this point, he's able to do photoactivation, photoinitiation of the reaction to get CO2 fixation and form the same types of products. The yields are a little bit lower, molar activity is improved, still not very high. This we published last year, and last year also Abhishek Patel joined our lab, and he's taking on this photocatalysis to move it further. He has a different approach where now he's going to start from carbonyl compounds, ketones, and aldehydes, and eventually to get to imines to form these kind of amino acids. Again, using a high molar activity approach and using photocatalysis to have good substrate compatibility. I won't talk about his work today, but he is talking in the whip rounds on Wednesday, so if you're curious about that, Abhishek will tell you about it at that point. Last project I'll mention is a different interest in amino acids. So from that previous project, we were able to make this compound, which is an unnatural amino acid. This is phenylglycine. And we do this by activation of either a CH bond or a CC bond at the alpha position. So this is interesting, and we liked amino acids. A couple of problems is that we required the phenyl group in that position. It would not work with other amino acids. And the second problem was that we required protecting groups on the amine in order to get this to react. So talked to Rylan about what the limitations of this method are, and we tried to figure out a way around this. And we thought about paradoxical phosphate and how nature does this. So what nature does is it takes an amino acid, reacts with paradoxical phosphate, and then in the presence of an enzyme, is able to affect this decarboxylation. Okay, so this is stuff that's going on all the time in the body. So we thought, well, can we turn that backwards? Can we get the decarboxylation to work and then flood in a bunch of CO2 and push this backwards and release the amino acid? So essentially do an exchange of one isotope for another. That was our hypothesis. So we started out testing pyridoxal phosphate as well as a bunch of other aldehydes. And again, this discovery process was done in Alberta with C13. And using a number of aldehydes, we do get incorporation. It's variable. It does depend on the aldehyde. Pyridoxal phosphate actually doesn't work all that well, but a very simple aldehyde, benzaldehyde, works quite well. And they have differing rates. There's some different kinetic profiles here that we don't need to get too deep into. But the catalysts are fairly stable if we use benzaldehyde or an electron-rich catalyst. And the reaction proceeds fairly quickly at the initial stages. So we thought this might be a method that we can use with amino acids. Now we're using unprotected amino acids, simply bubbling in CO2, elevating the temperature a little bit, and we get it to do the isotopic exchange. Moving this on to C11, we really thought about doing it in the catalytic method, and we found that preforming our imine, so taking stoichiometric benzaldehyde in this case, preforming into imine so that it's already like pre-activated, similar to the aminophosphate approach, then bubbling in CO2 and heating it up, we can get within 10 minutes a good amount of incorporation of CO2 to radiolabel these amino acids. The strength of this is that it's dead easy to purify these. All we do is we hydrolyze the imine, we end up with an amino acid, and then we simply do a little HPLC purification to get rid of benzaldehyde, essentially. There's essentially no other byproducts of this reaction. So we can get fairly decent yields for several amino acids, phenylalanine, leucine, some unnatural amino acids as well. So we're specifically interested in this leucine as we're interested in branched chain amino acid metabolism. So we've applied this as an imaging agent. I won't show you the data for today, but this is a preparatively useful method. We then thought, well, okay, this is the mechanism we think it's working by based on pyridoxal phosphate, is that we're getting this decarboxylation, recarboxylation step. And we thought, well, instead of carbon isotope exchange, then where we get low molar activity, maybe we can goose this, where we start with carbon dioxide on one amino acid, and then we add an aldehyde, we form these kind of aza-allyl anion intermediates, and then the CO2 can choose which side it goes to. So we start with phenylglycine, but we'll end up with leucine. So we'll call this a transfer amino carbonylation. And Maxime and Uzair were working on this, and they never observed transfer carboxylation. So eventually we had to go to Riley and say, hey, something wrong here. Mechanism is not what we think. And indeed, we then went about and did several other mechanistic studies that showed that it doesn't work the same way as pyridoxal phosphate at all. In fact, it's the reverse. So we do form our imine, and then we get carboxylation first to get this malonate intermediate, and then decarboxylation. So it's the exchange is happening in the reverse conditions, and therefore transfer carbonylation will never work by this method. But it means it's a very useful method for making radiolabeled amino acids, and we're able to enrich them going through these kind of malonate intermediates. And then we can focus on other ways we can do decarboxylation or break a different type of bond here that will allow us to access those in high molar activity. Okay, I will wrap up. So what I've shown you today is that we can use CO2 in many different ways for direct radiolabeling of complex small molecules. These methods do need to be designed specifically for C11. We always develop our methods with other isotopes first, and then inevitably we need to adapt them. We need to find ways to get this to work better with C11. So we need to start with C11 in mind for reactions that are going to be used. We can make reactive intermediates. CN bonds are easy to form. CC bonds are more difficult to form, and therefore we have to think about these two reactions in different ways and how we can make them compatible or preload our reagents so that one step is very fast and we can focus on another step. So photoredox catalysis is actually a major focus for the lab right now that Abhishek will tell you about on Wednesday. We're able to make interesting molecules, and we think several of these have utility in imaging, and we're using these as platforms for radiotracer discovery. This is the group as of last summer. Most of the work I've shown today was led by Maxime, Uzair, Braden. There he is. Now it's being led by Abhishek and a couple other students. Hunter also is leading our PSMA work. We were benefiting from a strong collaboration with Alberta. We are hosting a radiochemistry symposium at CSC in Ottawa. If you're interested in radiochemistry, therapeutic imaging, actually we're extending it beyond radiochemistry to all sorts of molecular imaging. Please come down to the Rogers Center now, I guess they're calling it. This is the lab as of last summer. This is the lab, we're turning over quite a bit. So this is the new group, and these are the funders. I'll borrow one more minute of your time and just put in a little promotion for our radiochemistry core, and that we're trying to offer new services. So we've recently hired a guy, Matt Dawes, and Christian is our lab manager. These guys are building up some capacity in organic chemistry. So if there is interest in any of these types of projects, not only doing petraceous synthesis and labeling drugs, but also doing some other chemistry that may be useful, they're eager to offer this as a service. So whether you're using fluorescent molecules or doing some small molecule development, purifying your reagents, they have capacity and they're eager to extend their offering. With that, thank you to everyone for your attention. I hope you've learned some chemistry today.
Thanks so much, Ben. That was excellent. I can't say that, I mean, I definitely learned a lot. I felt like I also got a little like PTSD, right? Which is pretty common probably where you'd go. I think you sort of just started to answer it there at the last slide when you described some of the applications. And I'm just wondering, thinking about, as I'm turning over in my head about where we could start to visualize and quantify reactions that we know are really, pathological. So thinking about your, marrying what you just described, the details of your chemistry, which is so important, how do we make those next level decisions about which of the steps, which of the enzymes, which of the processes, which of the, the kinetics of what we want to measure? How do we make those decisions? Like you've just convinced us that the chemistry, you can do it, you can figure it out. And how do you, so, not just which pathways, of course, that's the biology, but the properties of the pathways, the kinetics of the pathways, the molarity concentrations, the location, tissue, otherwise, just to kind of put that into perspective.
Yeah. I'd actually say that that's the question I'm asking more often, which is that what we need, like, it's fine and well for us to go and develop new chemical methods, and that's fun for us, it's stimulating for us. I love this stuff. But what we really need are challenges, right? So from my perspective, these all look like chemistry challenges to me. And that's exciting. That's the way we develop new projects. We need to know where our limitations are. So yeah, I mean, we come up with some of our own ideas, like, okay, I can read a lot about all this BCA metabolism that's so hot right now, and I think, how can I intervene in that? What can I measure there?
Right.
But we really need is the biological expertise to tell us this is what we think we're interested in, and then we work together to try to figure out how do we address that? Because really, biology is about molecules. So there's very few things we can't intervene in. I'd say one of the things we do need is a tissue location.
Yeah.
So imaging circulation is very difficult. Molecular imaging, because we don't have perfect spatial resolution, we need to be able to say, yeah, this is associated with a cell or at least an environment. In terms of the molarities and the pathways, what I'd say is typically things that are on the cell surface are easier. Things that are in the cytosol are easier than the nuclear envelope. But yeah, I mean, they're all addressable. What the concentration needs to be, that depends on the affinity of the molecule.
Right.
So it's also very helpful if there is a molecule known. Doing de novo tracer discovery can be very difficult. So typically we're often building these based on where's the medicinal chemistry going.
Right.
That might give us a hint that, yeah, there's something tractable there.
So that was going to be, but the next part of my question was the tracer development in your mind. You see it as sort of a partner to the medicinal chemistry, as opposed to the radiotracer being the molecule of discovery. Are you usually piggybacking off the medicinal chemistry?
It depends on okay, so if you're looking at an enzyme inhibitor, then yeah, you do need that because medicinal chemistry labs are much better resourced than pet tracer development labs. Often we're looking at what companies are doing, and they're able to throw a lot of workforce at it, and they'll come up with a library of large number of molecules. We cannot compete with that. We cannot replicate that.
Right.
So yes, but we have to keep in mind that a good drug has properties that are orthogonal to what's going to be good for imaging. So a good drug, you're going to take orally once a day,
Right.
and it's going to hang around in your body. And it's doing that because it's not so specific.
Mhm.
It may not be very selective. A lot of drugs are designed for polypharmacy these days. So we need to be aware of what the design parameters for them are and how we need to adapt that. How we may pick out from the same data set, we'll pick out a very different molecule from the ones that the medicinal chemists will pick out
Right.
for their development. On the other hand, I think there's a lot of excitement in terms of imaging biomolecules, right?
Yeah.
So if we want to study metabolism, then what better molecule to use than something that already exists in the body,
Yes.
or that we can modify slightly to make it like a locked
Yeah.
metabolite, right?
Yeah.
So there are ways to get at it. It's the medicinal chemistry is very helpful, but you have to be careful because drugs make poor tracers almost every time.
All the time. So the category helps you, but not necessarily the actual product itself. the
You
drug
understand the
itself.
core of it, and then you develop your molecule in a different way.
Yeah, okay. We have a question online from Dr. Beelands. Great work, Ben. Similar to my question. If a clinician or a scientist wanted to think about a new tracer target, what are the three things you think they should look at? And his examples are receptor, blood level, existing drug, whatever. So you're kind of answering it, but what are the three things you'd recommend?
Yeah, so it needs to be localized in a certain area. That's number one. Second is you do need a certain concentration. So in general, if you can expect that you have nanomolar affinity, then you need something like 100 picomoles of picomolar concentration of your of the target.
Mhm.
We're not typically, fluorescence can work with like a handful of cells. We're not working at that level just because we don't have the same spatial resolution.
Right.
So we need to be a bit of a larger target that way.
Yeah.
Third thing would be, is there, for me, the number one thing I tell my students is you have to have the application in mind.
Okay.
So know what it's going to be used for, know how this will be useful.
Mhm.
It's not, correlational studies are interesting, but we'd rather have something that is mechanistically important
Mhm.
and that is going to allow us to inform a decision from, and that's where I need to really have a clinical collaborator,
Right.
clinical collaborators
Right.
tell me this is useful or not, because it's not just about the biology itself.
Ben, wonderful presentation. For someone that doesn't know much about your field, I see a lot CO2 fixation and challenging in carbon-carbon formation. Have you or someone has considered to use the biological machinery that's already in place, for example, anaerobic bacteria, to feed the CO2 and try to get some more complex molecules?
Yeah, I haven't seen bacteria used, but people, and this goes back a while, but people were actually using plants. So if they want to make a series of sugars, they would simply incubate them with plants and then harvest them out again. It's tough to do. Just so nowadays, when you're doing clinical pharmaceuticals, you have to be working in a GMP environment and doing that kind of synthesis and having to grind up plants. We tend to like solution phase stuff a lot better now. There has been some done, and I think it'd be really interesting to do that, especially if you can develop it in like a flow format, right? So like if you can flow your CO2 over a bed of bacteria or plant cells or whatever it is, and then be able to extract out your metabolites, that would be very cool. I think it's a challenge for pushing it further beyond the discovery phase.
Yeah, great talk, Ben. Thanks. My question is thinking about if this CO2 fixation could be used to label cholesterol with C11 rather than like C14, for example. Would there be an advantage to using C11 cholesterol over C14 cholesterol? Would there be a narrower range of detection? So one of the things that we're interested in recently is doing like these dual reverse cholesterol transport assays where we pre-label cells either with tritiated cholesterol or C14 cholesterol. But there's overlap between the detection and the protocols with the liquid scintillation counting. So I'm asking not from the imaging point of view, but from just the radio label and having a radio label in-house versus the super expensive cost of it from purchasing it pre-labeled from Perkin-Elmer. Like, is this something that you could do in-house, and would there be advantages?
Yeah,
over
so
C14.
there will be energy resolution. So for reference, the photons we're detecting are centered around 511 keV, which is going to be, order of magnitude or more higher than what you're seeing with C14 or tritium. So we would be able to, you'd use a different instrument entirely in order to detect it. So there would be resolution. What I'd be concerned about is whether, because it's so high energy, it might also contaminate your signal for the lower energy emission. So I'm not sure about that. The other challenge will be the kinetics of it. So like, how long do you need to run the assay, and does C11 give you a long enough half-life for that? So we get this question a lot. Is there a way you can bring down my cost from Perkin-Elmer?
Yeah.
And I'd say that's a direction that maybe I'm hoping these guys can go in at some point.
Yeah.
We're not set up for that yet. But long term, I'm hopeful that we'd be able to do some tritiation or even some C14 chemistry.
Okay, very cool. Yeah, because like the C11 labeled cholesterol could also be like incorporated into HDL, into like infusion studies and things like that. And even the leucine, like I'm not sure, piggybacking off Marcelo's question, like could the leucine be used in bacteria for, ApoA1 synthesis, for instance, and then we'd have this like labeled A1 that we can, infuse into mice, kind of like the CSL 112, and see, the localization and uptake in the plaques. Do you think?
Yeah, I mean, it's possible. I'm just wondering, like, is that the best way to do it? Because it's just the challenge with C11 is the half-life, right? So getting enough out of that process to still run an imaging study is tough. There may be more direct ways to do that. And when you're talking about labeling, a larger biomolecule like that, you maybe have some flexibility in terms of where you can incorporate a label. And then we have, I haven't talked about it today, but we have a whole bunch of bioconjugation methods as well that are designed very well for that.
Okay, amazing. If no other questions, thank you so much, Ben, and thank you everybody for joining, and we'll see you at next week's meeting.